Good morning, everyone, or good afternoon, depending on where you're joining us from. Welcome to the fifth International ADTKD Summit. ADTKD, of course, stands for Autosomal Dominant Tubulointerstitial Kidney Disease. Now, ADTKD, as you may or may not know, is the second most prevalent genetic kidney disease in the U.S., second only to PKD. It's estimated that between 75,000 and 100,000 individuals in the U.S. alone suffer from this disease, and it could be as many as 2 million worldwide. But because of its non-specific clinical presentation and subsequent underdiagnosis, these numbers could be even higher. So my name is Sharon McBroder. I am the Executive Director of the Rare Kidney Disease Foundation, or RKDF. I'm also an ADTKD patient. I have the MUC1 variation, and I am currently in stage 4 pre-transplant. I have watched needless suffering and early death among my mother, my grandmother, my uncles, dying from renal failure over the years. And before we knew it was genetic, I suspected it was genetic. It was just, we came from a large family. So the odds that, 4 and 6 siblings have this disease struck me as kind of odd. But it's because of my family history that I first started volunteering for RKDF. And in December 2024, I became its executive director. Or I guess 2023. Time flies. In fact, RKDF has all the staff have or have a family member with ADTKD. So we understand the urgency around finding a treatment and for this currently incurable disease. So RKDF's mission is to inspire hope by working to halt ADTKD across generations through education, patient advocacy, and community building. And so that's why we're here today, to learn, to hear from ADTKD patients, and to build the ADTKD scientific community. So welcome. We're glad you're here. First of all, the summit would not have been possible without the hard work of RKDF leadership team, including Sue Real, Suzanne Kelly, Kathy Stankus, Lisa Parcell, and RKDF co-founder and president, Richard Nelson. Nor would it have been possible without the social media and advertising efforts of Jess Leong, Christine Upton, Joe Morden, and Joe's fantastic team at TMV Marketing. So thank you all. Just a program note for today, Dr. Anna Greca's presentation on therapeutic advances in ADTKD will now be at 10:50 a.m. and Dr. Maria Pia's presentation on calorie restriction and UMOD progression is now scheduled for 11:25. So we just switched those two sessions due to scheduling constraints. All right, a few housekeeping items. First of all, you'll use the same Zoom link for today and tomorrow. So if you made it here today, great. You just need to click that link again for tomorrow. Attendees will be muted throughout the entire summit, but we do want to hear from you. So after each session, we will have between a 5 or 10-minute Q&A session. You can submit your questions through the Q&A Zoom functionality there at the bottom. And when you're posting your question, you will have the option to send anonymously if you wish. And so there's a box there you can check to just say send them anonymously before you post it. Now, if your question is aimed at a particular panelist, please mention that, and we'll make sure that they can answer your question. And if we don't get to your questions live here during the summit, we will do an FAQ or a Q&A afterwards. We'll put out one, and we'll make sure that we get expert answers to all of your questions, and we'll send that out and post it on our website. And please note, we are not able to answer personal medical questions during the summit. That is not our role and far from our expertise. If you are having any technical difficulties during this program, just email info@rarekidney.org. And Suzanne will take care of you. And then just another note, as you heard, we are recording this summit And parts or all of the sessions will appear on RKDF's YouTube channel. We have a ViewMedi channel, that's VUMEDI, and part or all of the presentations will be posted there. And then snippets will be used in our social media posts and on our website. So just so that you know, this will be publicly shared. Okay, so let me tell you what we're going to do today, first of all. During today's science program, you will learn about strategies for diagnosing rare genetic diseases, including ADTKD. You'll learn about cutting edge research on UMA disease management through calorie restriction, groundbreaking therapeutic advances in ADTKD, and the nuts and bolts of how to integrate genetic testing into your clinical nephrology practice. Now, we're going to anchor this summit in the voices of ADTKD patients and family members to remind ourselves that that's why we're all here. We're here to learn about ADTKD, but we as the patient organization and our patient volunteers, we want to convey the urgency in finding a treatment for this currently incurable and often invisible disease. So let's turn to our first session. Patient voices. We'll begin with Cynthia and Cliff. Let's see them join us here, and then I'll do a brief introduction of them. Hello, Cliff.
Hello.
And Cynthia, welcome. Thank you for joining us. Cynthia is an RKDF volunteer. She's a member of our clinician council and a retired pediatrician. She has the MUC1 gene variant of ADTKD. Her brother Cliff is a soon-to-be retired aerospace engineer and also has MUC1. Before I turn it over to them, I just want to remind you, if you have questions for Cynthia or Cliff, please put them in the Q&A below. All right, Cynthia?
Hello, everyone. I'll try to share my story in a concise way, and then Cliff is going to finish up our family story, I think, and then I may chime in again later. I wanted to, not to make things too complicated, but I wanted to say that we were pretty lucky in that ADTKD affected our family, what we know of the family who has it, relatively late in life. So as young kids and children, we were lucky to be free of this disease and of the knowledge of the disease. But now we know so much more, and I wanted to thank the organization for that. But I did know that I had some kind of kidney disease very early in life. I was getting routine checkups, and my urinalysis showed that I had some red blood cells in my urine, just a small amount. So I was referred to a nephrologist at about the age of 10 or 12. I can't remember exactly. And I had some evaluation and an intravenous pyelogram, which was the standard of diagnosis at that time, and it showed normal kidneys. So given that red blood cells in my urine were the only finding, I was diagnosed with probable thin basement membrane syndrome, which I was told at the time that was a benign condition. My mom probably also had it, and one or two of my brothers had it. And they were told would never cause any problems. I didn't need to worry about it. So I put it out of my mind, just something that I put on applications and medical forms and so forth when I got jobs and things. But then when I was in medical school in my 20s, my dad started having elevated creatinine, and initially his primary care doctor was not concerned, but eventually he did get referred to a nephrologist. And I remember attending one or two of those appointments with him, and my dad was otherwise very healthy, never had any problems, no diabetes or hypertension or anything like that. And I asked the doctor, what's the cause of this kidney disease? And he said, oh, probably hypertension, hypertensive nephropathy. And that didn't really satisfy me because my dad had never had hypertension. Like I say, he was otherwise very healthy, didn't take any medications at the time. So anyway, he went on to eventually require dialysis, but not until his 80s. And he was very healthy. He was on dialysis for 10 years before he died at the age of 94. But I was frustrated by that lack of diagnosis. And of course, in the back of my mind, I was always thinking, is this something that we could inherit in our family? Um, and I did know that my dad had a first cousin who had died of kidney disease in her 40s on dialysis. Nobody knew why. There was never any diagnosis or any knowledge of any other people in our family that had it. Um, and then in my 40s, my creatinine started to rise a little bit. And again, my primary care doctor was not concerned, probably just dehydration, don't worry about it. But I told him about my family history, my dad, and insisted that I get the tests repeated, and they were worse. And I was referred to a nephrologist at that time, and was followed for many years by this nephrologist who was understandably concerned but confused because he couldn't come up with a diagnosis for me. I had many, many tests, and eventually, when I changed insurances in about 2016, my new nephrologist said, "Oh, you need to have a biopsy, first of all," because I hadn't had a biopsy yet. And of course, that biopsy showed thin basement membrane disease. So we kind of settled on that as my diagnosis. But again, I was confused and unsatisfied because I had been told that was a benign diagnosis and shouldn't cause kidney disease. And when I pressed my nephrologist about it, he said, "Well, it is in this case. It's causing cell death in some way that we don't understand." So I was, you know, I sort of settled in, and that was the case. And I was a little taken aback by his lack of interest in finding the real cause. I mean, what I thought should have been another cause, because there was, well, there was no knowledge of this gene, at least by my doctors at this time. I think it had just been discovered by Dr. Blyer. And and again, the diagnosis I was giving just didn't make sense to me. So that's when things started to overlap. I had another brother, our youngest brother, who started to have kidney disease at this point, and he, at this time in 2016, he was already about stage 3, stage 4, and he hadn't had any knowledge that he was losing kidney function until that time. And then I think Cliff started having problems about this time, and I'll let him take it from here, and he can tell you what happened next.
Okay, thanks, Cindy. Yeah, and just going back to the earlier time, I was probably less involved in my dad's issues with his kidneys. I mean, I knew about it, maybe not as early as Cindy did. And it was probably about 20 years ago when I was in my 40s, also early 40s. I think when I first had a high creatinine reading, and the doctor said, "Well, that's a little unusual. Maybe you were working out, maybe you were dehydrated. We'll do it. We'll take another measurement." But no real concern. And just over the years, going to physicals every year or two, and every year the creatinine was up a little bit. It's very, very slow progression. Again, my primary care doctor wasn't too concerned about it. I mean, he was monitoring it. And I knew about Cindy's issues, and of course, as well as my dad's by that point. So this was probably 2017 or 2018, I think, when I said, well, I should probably start seeing a nephrologist. And he gave me a referral to the nephrology department. I picked a nephrologist and saw him a couple times. Again, there wasn't a real diagnosis. My blood pressure was a little bit elevated. He gave me the advice, just drink plenty of fluids, try and drink 2 liters a day, avoid red meat, things like that. And a year or two later, he retired, and I got a new nephrologist. And well, I guess I should mention too, at the time I first saw a nephrologist, I told Cindy about it, and she told me about our other brother Ken, who also had kidney disease. I had no idea at the time what his issues were. So, so I think I let the first nephrologist know that. He didn't seem too concerned, but then when I got the new nephrologist and told him about the family history, he looked up Dr. Blyer's papers about the different genetic causes for kidney disease, for ADTKD or for TKD. And at that point, too, I had also had a kidney biopsy before him with the old nephrologist, not on his suggestion, but just because I'd known Cindy had a biopsy, and I thought maybe we'd learn something from that. It turned out that the interpretations of our biopsies were a bit different. I had nephron death and interstitial fibrosis. Yeah.
Mm-hmm.
So there wasn't too much there, but once I had the new nephrologist and sent my samples off to Wake Forest and to the Broad Institute, that was in 2019, in around April or May. And then in the meantime, our dad passed away that year in September. And so after that, I started asking, have we had any results back from this testing yet? And I think it was in October that I got the call from Dr. Blyer, found out I had MUC1 disease. So since then, kind of just been dealing with it. It's just hanging over my head. It didn't have a big effect at first. It was certainly nice to know what was going on, and then to be part of RKDF and the community, meet other people with the same condition.
Yeah, and I can add that as soon as Cliff got his results back, Dr. Blyer said we need to test the rest of your family. So I was tested,
Yeah.
and my mother was tested. She was still alive, and my two daughters were tested eventually. And my mom does not have the condition, so we assume that it came from my dad. And my two daughters both have it as well. So it was just such, I have to say for myself, such a relief to finally have an answer and to know what was causing the disease, even though there's no cure. We at least had some sense of closure, at least I did. And I'm really grateful for that. And I wrote back to my old nephrologists, both of them, and my primary care doctor, and I told them exactly what was going on and hopefully helped educate them a little bit too. And I've had a transplant now in September, and doing very well, thank goodness. And thank God for the living donor transplant program for the National Kidney Registry that got me a transplant within 3 weeks of signing up. And I was lucky to have a friend who was willing to donate on my behalf. So I was very lucky. And my brother Ken had a transplant in 2020, and he's doing very well as well.
Yeah,
And
and I'm probably on track in a few years. I'll probably be next.
Yeah.
Right?
Yeah. And I'm really motivated to find some kind of treatment for this disease because both my adult daughters have it, and my one daughter has two children who haven't been tested, but very good chance that one of them could have it. So I think that's all I have to say about you, Cliff.
Yeah, I just want to say too, I'm grateful for the all the researchers and doctors and scientists who are working on this. So. And it's been great. Met some of them at the Kidney Week, American Society of Nephrology. Is that right? In San Diego a couple of years ago. So.
And you had the chance to visit the researchers in Czechoslovakia too when you were there, right?
Yes, we did. On a trip to Prague, I did meet Stan Kmock and Martina Zivna, and they showed us their lab, and so they were very, very nice. It's a great time.
Yeah, and I was lucky to be able to visit Dr. Blyer at his lab as well a few years ago. That was really fun.
Well, thank you so much for sharing your stories. Um, I didn't know you had met Stan and Martina over there in Prague. That's amazing. They're doing such amazing work along with Dr. Blyer. Um, I just had a question, and then we'll see if there are questions from the audience. Given the stage you are each now, so you're pre-transplant, Cynthia, I'm guessing you're then stage 4 or 5.
No, I had a transplant in
Oh, you
September.
had just this past. Okay,
Yeah,
gotcha. So
yeah.
how are you feeling? This is my question. How are you feeling?
I'm feeling good. Great. Yeah. Yeah, some minor side effects from the medications, but I'm feeling I have more energy and sleeping better and just generally, I mean, psychologically, I'm feeling much better.
Uh,
Just it's great to get it over with and be on the upswing instead of the downswing.
yeah, I understand. And Cliff, how about you? So you're pre-transplant. Are you
I am
stage
pre-transplant,
4?
stage 4, yeah. So yeah, I mean, initially, really, I was in good health, didn't feel anything, but in more recent years, definitely feel more fatigue. I've had other medical issues, having to take a lot of pills. Some of it related to the kidney disease, some of it probably just related to getting older.
You've had
But
gout too, associated with it, right?
yeah, gout's one of those. But yeah, really, the thing I've noticed, I think, is a little bit more fatigue than normal. Out hiking or working in the yard, or it just can get fatigued very easily. Yeah, and yeah, another thing, a little bit of temperature sensitivity as well. I chill
Mm-hmm,
easily,
mm-hmm.
and I
Yeah,
think
Well, and I
that's
can relate to all of that for
yeah.
sure. Yeah, I have my little heater down here in the basement in case I get cold there. But well, let me turn it over to Lisa to see if we have any questions from the audience.
We have a scientific question, but also a comment, and it says, sounds like your family has almost all inherited the mutants. Very sorry about that.
we have one brother who's unaffected. Yeah.
Yes. And Sharon, so the rest of the question is more for the science side, so I'll probably save that for later.
Okay. Okay.
And that's
Yeah,
it for questions.
related to that, I could mention that I've done a little bit of genealogical research, and it doesn't seem like there's any early deaths in Dad's ancestry, really. I
Well,
mean, his
except for that
parents.
one cousin.
Yeah, the one cousin.
Yeah. Yeah, so I'm in the process now of drilling my cousins that I do know to try to find any other relatives that we might have in our extended family that
Right.
might have had kidney disease, particularly people who are related to that one cousin of Dad's. But I'm not getting very far. We lost track of people. But no, we haven't heard of any others. No.
And I've turned to Ancestry to see and death records and things, but even that, um, either they don't know that it was kidney disease. When you go back far enough, it's not going to say that was the cause. And
Yeah.
so at that point, it really is, was it an early death or, and I came from a large family, so you do have the opportunity to see more people in a single generation, which is a
Hmm.
clue. But it really is hard, I think, to track back because they just don't know a lot of times what was the ultimate cause of death.
Hmm.
Or,
Or they
you know,
had kidney disease, but it was due to something else because they were
right.
89. Yeah.
Right. Well, if we have no more comments or questions for Cynthia and Cliff, I just want to thank you again for joining us today. Um,
I'm sorry to interrupt. There is another question and it's a great one. So do you mind if I ask them really
oh,
quick?
please. Yeah.
Okay. It says, I'm curious if you had genetic counseling before or after getting tested. If so, was it helpful?
Uh,
I did not. Yeah.
I did see a geneticist at one point, but truthfully, she wasn't able to tell me as much as Dr. Blyer told me. And he was the one who was the best counselor for me in terms of helping me get the rest of my family members tested. I mean, there really isn't anything that can be done, and I don't think there's any prenatal testing yet. So, no, I didn't really have any formal genetic counseling.
What about the question?
And just to follow up on that, would it have been helpful? I mean, had you known it was available before, during, after, do you think that would have been helpful to talk to someone?
Sure. Sure. I mean, maybe we would have had an earlier diagnosis, but again, that wouldn't make a lot of difference in the eventual outcome. I think once there's a treatment or some kind of medication, it'll be much more important, especially if it's something that young people can take.
Yeah. All right, Lisa, any other questions?
No, that's it. Thank you.
Thanks. Well, thanks again, Cliff and Cynthia, for for being here, for being part of the foundation. You know, we couldn't do what we do without, dedicated volunteers like you, people committed to the cause, because unfortunately you've experienced it personally. So again, I thank you very much.
You're welcome.
Yeah, thank you.
Okay. All right, next we have Martina Zivna. And Martina holds a PhD in molecular biology, genetics, and virology, and is currently a senior scientist with the clinical research team led by Professor Stan Kamak, we had just heard, at the Department of Pediatrics and Inherited Metabolic Disorders at Charles University in Prague, Czech Republic. She participates in the international multi-site project aimed at identification and characterization of genetic factors contributing to inherited tubulostitial kidney disease, including REN, SEC61A, UMOT, MUC1, ALG5, and APOA4. She recently developed a non-invasive urine-based immunolabeling technique for diagnosing MUC1. And last year, we are just so proud and excited for Dr. Zivna. She was awarded a prestigious 4-year grant by the Czech Minister of Health to continue her research in the field of inherited tubulointerstitial kidney diseases. With that, thank you for being here, Martina, and I'll turn it over to you.
So, hello. I will share my screen. Can you see my screen? Can you see my screen?
Yep.
Yeah. Okay. So, good morning for US. Good afternoon for Europeans. So first, I would like to thanks to organizers to give me the opportunity to present in this wonderful conference topic about diagnostic strategies for rare kidney diseases. I joined the team, the Prague kidney disease team in 2002 as a PhD student when I joined the team of the Sten Kmoch. And we are focused on identification and characterization of genetic causes of rare kidney diseases. In these days, we are clinical research team together with the team of Dr. Blair and Kendra Kitt, and we also worked very closely with other nephrologists and groups such as Irish group of Peter Conlon. So how we are working? Based on medical history and symptoms, we are collecting set of patients and families with a specific disease. First, we analyzed DNA to identify the causal gene and mutation for the disease. If we identified this mutation, then we can make a genetic diagnosis. And additional to the genetic diagnosis, we also aim to identification and characterization of the biological consequences of mutations, causal mutations, and the knowledge of genetic diagnosis and molecular mechanisms of the disease is basis for the therapy development. The universal biological concept proposes that genetic information encoded in DNA is translated to protein through transcription to RNA. Protein synthesis occurs within cell, and it's responsible for physiological functions of the cell organs and entire organism. In a human body or human DNA, there is around 20,000 genes coding proteins, but it's only around 2% of entire DNA molecule. And in health, all of these cascade is fully functional. If some of gene is mutated, it may lead to production of the protein that is ill functional. And such a protein may either not be included in the physiological functions and cascades, or may be toxic for cells that produce it. The disease type and severity depends on the type of cells that produce mutated protein, or physiological functions that is responsible the affected protein. Now let me present hypothetical cases that we are solving within as a part of our genetic kidney research. So you can see different type of inheritance and clinical and biochemical presentations of the typical signs from de novo development of disease through recessive type of inheritance to dominant type of inheritance. And in such some of cases, we the medical history or family history and clinical signs of the disease are not informative, and the genetic disease or genetic nature of the disease is questionable. In these cases, we consider genetic disease when the cases kidney fails, or in individual kidney fails before 50 years of age. In ancient ages of genetics, the solving of cases was based on linkage analysis and then subsequent single gene sequencing in or from affected DNA region. For example, in family with the dominant type of inheritance and cysts was very often analyzed PKD1 gene, which is responsible or associated with polycystosis of kidney. In these days, the routine genetic diagnostic lab use clinical gene panel that allows to analyze 200 to 300 genes that are associated with kidney diseases, or they can use whole exome sequencing that allows to analyze all parts of DNA that encoded proteins, which is around 20,000 genes, but only 2% of entire DNA. And there is another approach which we often use, and it's analysis of whole genome sequencing that is analysis of entire DNA molecule. And there are three types of responses from routine genetic lab or genetic diagnostic lab that the nephrologist can obtain after the testing. The first type of response is positive, is the best for patient because the pathogenic variant was identified and the genetic diagnosis can be made. And if possible or if available, nephrologist can start with the specific treatment. The second type of response or result is inconclusive. It means that suspicious variant in a known gene to be associated with kidney disease was identified, but there is no biological evidence for this specific variant and disease development. These variants are known as variants of unknown significance, and it needs our additional experimental tests to close the diagnosis. The worst case scenario is negative result. It means that no gene variants meet the criteria of type of inheritance, type of disease, and population frequency was identified. In rare genetic kidney diseases, the most often result from the routine diagnostic lab is either inconclusive or negative result. And these cases are cases for our team, for our lab. So now I will show you two examples of how knowledge of biology can help in accurate genetic diagnosis. First case is a case of Australian family that clinically fits to tubulointerstitial kidney disease. The proband in the family pedigree in the red square was analyzed by the routine genetic testing lab, and one of the reported variant was UMOT variant. But this variant that affects structure of the uromodulin at the position 55, position amino acid. But this variant was classified by the bioinformatics as a benign, which means not pathogenic, not related to the disease. But the nephrologist contacted Dr. Blair with the question if this variant could be pathogenic for the disease. And Tony Blair referred this family to us. So what we know about uromodulin? Uromodulin is produced by a very tiny part of kidney by a specific cells that called tubular cells, and these cells processed uromodulin, which is exposed on surface of tubular cells, some of the tubular cells, and from where it's released into the urine. However, mutated uromodulin that cause ADTKD is not properly processed by the tubular cells and gets stuck inside of the cells, is not able to reach membrane of the cells and is not released into the urine. So we started with the analysis of uromodulin in kidney biopsy from patient with investigated variant, and we found that uromodulin is retained inside of tubular cells and is not present on plasma membrane, which corresponds with absence of uromodulin in the patient's kidney, in the patient's urine. So we concluded that formerly benign variant, we reclassified this variant for pathogenic based on the results that UMOT is retained in the kidney and absence in the patient urine. So we solved the case of this family as ADTKD due to UMOT mutation. And this family is now included in the registry of Dr. Blair. So the second example or second case which I would like to show you is a family with, again, ADTKD, autosomal dominant interstitial kidney disease, which was referred to us directly from Dr. Blair and Kendra Kidd. In this family, the routine testing lab, genetic testing using clinical gene panel was negative. And Professor Blair and Kendra Kidd sent to us the samples. And by the whole exome sequencing and whole genome sequencing that we did, we identified variant in ApoA4 gene that have never been associated with development of tubulointerstitial kidney disease before. And the staining of ApoA4 in a kidney biopsy from patient from this family was not informative. So we didn't know if this variant could be pathogenic, could be cause of the disease, but within next 2 years, through the registry of Professor Blair and Kendra Kidd, we identified additional 4 families with another variant in ApoA4. But we still couldn't find the biological mechanism why in these families is developed chronic kidney disease and kidney failure until one of the patients was subjected to nephrectomy, and we analyzed whole kidney tissue, and we found or detected the pathogenic protein structure in a very deep part of kidney, which is not typically included in kidney biopsy. And these structures, pathogenic protein structures called amyloid, and these structures are formed mainly by mutated ApoA4 protein. So we can conclude that in these families, we identified a novel genetic cause of ADTKD, autosomal dominant interstitial kidney disease, and we also identified the biological mechanism of the failure and kidney damage in these families. So by our approach, which is a combination of genetic testing and identification of biological consequences and molecular mechanisms of the disease development, we significantly contribute to building of the registry with TKD cases of Professor Blair and Kendra Kidd. In this registry still remains around 70 families with ADTKD and unknown etiology. And we are testing 6 novel genes potentially associated with this disease. So we can conclude that genetic cause of rare hereditary chronic kidney disease is solved by routine lab testing from around 40%. Cases with inconclusive or negative results can be solved by clinical research team as we are. And knowledge of the basic biology of causal proteins is an important step to solve genetic diagnosis. Accurate genetic diagnosis of the disease is important for genetic counseling, prenatal diagnosis, selection of a healthy kidney donor within the family, and therapy development. So thank you for your attention. And my thanks also go to my colleagues from Research Unit for Rare Diseases in Prague, and also to the team of Tony Blair and Kendra Kidd from Wake Forest School of Medicine. So thank you for your attention.
Thank you so much, Martina. That was so interesting from what I could pick up anyway. But to see the extent of and your process of how you go detective work to figure
Yes.
out when there's mysteries and the what's available in the research lab to dig deeper, all in the effort to understand this disease, find out the biological pathway, and ultimately find a treatment. So thank you so much for you and your colleagues for all the work that you're doing.
Mhm.
Let me turn to Lisa to see if we have any questions from the audience.
Hi, yes, we do have a couple actually. In the first
Yes.
question, Martina, it says, in case number one of Dr. Zivna's talk, is the patient heterozygous for the UMOD variant? If so, is the urine analysis for UMOD only for the pathogenic variant, or is it for all of UMOD?
The urinary test can stratify the variant in UMOD to benign and pathogenic. The pathogenic in case of pathogenic variant, the UMOD is absent, and it's a specific for the UMOD ADTKD UMOD or patients with the UMOD pathogenic UMOD mutation.
Wonderful, thank you. Another question. In the case example where the variant was reclassified, can you clarify if the overall classification was benign or just in, is it silico predictions?
This classification is based on some criteria which combine the position of the mutation or variant, type of the change, and how this change affect the protein structure. It's in silico calculation. And there are some criterias which is called ACMG criterias. According the bioinformatician evaluate the pathogenicity of the variant. So it's in silico, and if the variant is, I would say, detected for the first time, then it's only in silico. And if you don't have any other tests, I would say experimental test. So, for example, when is the family with TKD, with tubulointerstitial kidney disease, and someone identified the variant in UMOD, and this variant results in amino acid exchange, then I think that always is important to test if this variant is pathogenic or benign, because in silico calculation, it's still calculation in computer, but the biology could be different. But it's the routine genetic lab sent to nephrologists some, I would say, recommendation, and they are not continuing with these specific tests, because these urinary tests, for example, are very specific, or it's not the routine expertise. But it's not a problem for us if someone referred family with this question, if the variant could be pathogenic. So we always analyze urine if available, or kidney biopsy.
Thank you so much, Martina. I don't know if there are any more questions. If there are, we will follow up and have Dr. Zivna finalize answer them, and we'll send them out. But thank you for joining us today, and we'll move on to our next session.
And thank you for the invitation. And I would say also, I would send greetings to Cliff Sedlund, because we met in Prague half a year ago. Yeah, it was nice.
Very good. Thank you so much. Okay, moving on now. It is my great pleasure to introduce Dr. Anna Greca. Dr. Greca is a core member and director of the Kidney Disease Initiative at the Broad Institute of MIT and Harvard, where she serves on the Institute's executive leadership team. She was also a professor at Harvard Medical School and a physician in the Department of Medicine at Mass General Brigham. And we at the foundation are just so grateful to Anna and her team for all that they have done in researching the biological pathway, which I'm sure she'll talk more about today, and really setting the groundwork for finding treatments. And so we're just grateful for her team's work. She also founded in 2024, I believe, the Ladders to Cure Accelerator, and this is to accelerate advances leading to treatments and cures for patients with all different kinds of genetic diseases, given the research on what she calls the nodal biological pathways. So anyway, without any further ado, I just want to get right to Anna's presentation. And again, thank you, Anna, for joining us today.
Thank you so much, Sharon. And thank you to everyone at the foundation and all the colleagues who are here today. It's really an honor and a great pleasure to be with everyone and to be a part of this summit. And I just want to take a moment to acknowledge just how amazing it is that we're all here together for these next couple of days. It's not so long ago that, there was just a handful of us. And now this community has just grown by leaps and bounds. And it's just so wonderful to see that because it really takes a village to get to where we want to go. And so I'm just really so very pleased to be a part of all of this. These are my disclosures. It is relevant that I will speak about our collaboration with Alexion and AstraZeneca as part of our discussion today. So I just want to start with the idea that we're actually living in what I call a golden era in biomedicine. We have the ultimate goal of preventing, curing, or treating, managing all diseases for all patients. Of course, as a physician scientist, this is my ultimate goal for my career. Of course, I'm slightly partial to ADTKD and all of you patients, with these diseases and just trying to make headway in this space. But I just want to acknowledge the fact that, obviously, I liken our work to climbing a mountain because it is a very long and arduous process. Sometimes you go up the trail, sometimes you have to stop, sometimes you have to backtrack and make your way back up again with new plans. And I think many of you have followed this path for a long time know that it's not a straight shot, to where we want to go. And yet, we have never had more tools available at our disposal in order to get there. And so I just want to say that, we have small molecules or pills, if you will, that allow us to move forward in this area. And I think we've made headway there. We have next generation treatments, including, for example, ASOs, antisense oligonucleotides. I will speak very briefly about that today. So we have additional backups, if you will, additional strategies. And there is an opportunity for a cure, which would mean to go inside the kidney, in the kidney cells, in the nuclei of those kidney cells and fix the DNA misspelling that causes these diseases. We are far away from that. I just want to make sure that everyone hears me say that. But nevertheless, it's not science fiction. We have the technologies to be able to do that. It just will take some time before we can, safely deliver them and efficiently deliver them to the kidney. But we do have opportunities that go from managing the disease with the pill all the way to a cure. And I think that places us in this golden era in biomedicine where we have all these opportunities available for us and for the next generation, if you will, which I know many of us here care deeply about. My group is interested in membrane proteins. We're interested in human genetics and membrane proteins and how disruptions in these proteins lead to disease. And I'm just showing you here, in addition to MUC1 and UMOD, which of course everyone here understands and recognizes, some of our prior work has also been in membrane proteins. And the reason I say this is just to create some reason to believe, if you will, that we can harness our understanding of these diseases in order to develop treatments and cures. And so just as an example, my group discovered the work that went into targeting the TRPC5 ion channel for a different set of diseases. And we reached all the way to phase 2 trials. So phase 1 is safety, phase 2 is efficacy. We reached phase 2 trials in patients with that discovery from our lab. And this is now a project that is with Bristol Myers Squibb, and we'll see if we can make it to the pivotal trial. But I'm saying this to say, while this is not related to ADTKD, that we have experience bringing treatments all the way to the clinic. And this is exactly what we're aiming to do here. Even if the path is not straight, we are absolutely determined to get there, and we have done it before. MUC1 and UMOD ADTKD diseases. Okay, so how do we get there? I just wanted to present a little bit of background as to what motivates this concept of nodal biology that Sharon briefly referred to, and this founding of the Ladders to Cures Accelerator, because it's relevant, very much relevant to ADTKD, and I would love all of you to be a part of this idea. So the first thing to say is the discovery and development of new treatments is a risky process, and it's difficult. It's, again, like climbing up the mountain, as I said. And I hope that it wouldn't surprise most of you to learn that, unfortunately, even today in 2025, the likelihood of going from a first in human, so from the phase 1, the first in human study, all the way to approval remains at 13%. This is probably one of the most failure-prone human enterprises that one can imagine, drug development, right? So it's a very long and difficult process. And even with all the tools, this golden era that I just made the case for, we still have a long way to go. And there are, of course, more and more tools coming in line that are available to us. But I just want us to, maintain a level of humility about the task that is ahead of us. We want to get all the way to approval. The odds are not in our favor, and we have to take multiple shots on goal in order to get there. So there are many reasons for these failures, but one of the reasons that I would like to discuss with you today is that our traditional approach of taking one disease, identifying one target, and then developing a singular drug for that target, this linear approach, if you will, is one that, of course, makes it difficult for us to get to success, because if we fail to make the right prediction at any point along this path, this singular path, I think that gives us fewer shots on goal for the different diseases that we are targeting. So what I would like to present, and obviously Albert Einstein said, in the middle of difficulty lies great opportunity. I see this as an opportunity. Someone could say, oh my God, these odds are so disheartening. What can we do? But I do think that we have an opportunity. And the opportunity, one of the opportunities that our team has spearheaded is this concept of a biological node, of nodal biology. So a biological node, we define it as a point of convergence on a single druggable node, the druggable target, that enables the discovery of connections, previously unseen connections between diseases that may have nothing to do with one another, and yet now they're connected because they share an underlying mechanism. So we're not going by symptoms or organs, but we're going by biology, the underlying fundamental biology. And of course, with the advent of artificial intelligence, we can actually use these tools to identify more nodes. And that's part of the mission of the accelerator that we have built at the Broad. But all of it was inspired by ADTKD. And the idea that we found that there's a shared druggable node that is not only shared by MUC1 and UMOD, but by many other proteinopathies. These are diseases that are caused by misshapen, mangled proteins that accumulate inside cells, but not only in cells in the kidney. They also accumulate in cells in the eye, for example. And so we're also working on a form of blindness that runs in families, just like kidney disease runs in ADTKD families. And we're also working on a form of Alzheimer's disease that runs in families, and other diseases that are also similarly genetically defined proteinopathies. And this TMED cargo receptor is a node that can serve all of these diseases in a way that is completely unpredictable. No one would have thought that an eye disease and a kidney disease would have something in common, and yet they share this same underlying mechanism. And so we have the opportunity to develop new drugs, not necessarily a singular drug, but multiple drugs for all these different diseases, but by targeting the same molecular node, which actually now gives us more shots on goal for all of these diseases, right? So this is the concept of nodal biology that I wanted to introduce. In the case of ADTKD, again, with the advent of artificial intelligence or machine learning, this is a structure that was predicted by the computer for this druggable node, the cargo receptor node that I just showed you. What is in orange is a particular component of this node that we are very, very interested in because it's an opportunity for us to develop a very specific drug for this node. But I just want to make the comment that by the combination of artificial intelligence and all of our prior understanding of the biology, we now have these incredible tools that allow us to build bespoke drugs, if you will, that just so will fit into the right place in this structure that you see. To you, it may seem like ribbons dancing. For those who are the scientists here, it will make perfect sense. It is the structure, it's the multimeric structure of a thymic cargo receptor complex. But the point is that this AlphaFold structure gives us an opportunity to develop the right drug for this target. And as many of you know, again, I mentioned to you, drug development is a very risky process. The more information you have, as we now have, the better drug we can build. And so, we have a new compound that fits just so into this pocket. We understand much better how to deal with this. This is information that allows us, to move this project forward and avoid prior, liabilities or toxicities, if you will, that these small molecules may have had, so that we can actually build better compounds. So all of this knowledge, including the structural information that we now have, allows us to build better drugs. And so I just wanted to show you using not the new compound, which I can't say much more about, but more to say that we're using the tool compounds that we have previously developed at the Broad. I can show you how well it works in the kidneys of animals that have MUC1 and uromodulin kidney disease, to give you a reason to believe that we now have everything we need in our armamentarium to build the best possible pill. We don't have it yet, but we're building the best possible pill to address these diseases and to put it into phase 1 trials as quickly as possible. And so this in green shows the tubules of a knock-in MUC1-FS mouse. These tubules are not normal, just like in the patients. As you can see in the biopsy here, these tubules are very wide. It's called tubular dilation. If you give the drug for 3 months to a mouse that carries the human MUC1 mutation, then you see that there are these big dilated tubules shown here in this orange circle. If you give the drug, the tubules become, we're able to prevent the tubules from becoming so wide, is a better way to say it. And if you look at the control animals, so these are the animals that do not have the mutation, you can easily see that the tubules of the treated animals, where we have prevented this dilation from occurring, I want to make sure that you understand that these are all shown at the same magnification. This is not a zoomed in picture. This is how big the holes are, if you will, or the dilated tubules. But this is where we're able to mitigate that from happening so that they look much more similar to normal control kidneys. So if we could do this in a human kidney and we can prevent this from happening, we would obviously have a very, very attractive treatment for this disease, right? So very much interested in advancing this knowledge toward clinical trials in patients. Now, what about UMOD? I want to make sure that we address both diseases. This is a mouse model that carries one of the UMOD mutations, as shown here. And I'm showing you here data that we published about a year and a half ago, where we basically treated these animals with the tool compound. And as you can see, we have a very significant reduction in the mutant protein in these animals. This is with a mutant-specific antibody that we were able to raise for this mutation. And we're also able to reduce the target of the drug, the cargo receptors that we're looking at. So we're reducing both the misshapen protein and the cargo receptors that hold on to it inside the kidneys. And so we're able to mitigate the disease in the case of UMOD as well, using the same nodal approach, right? This is showing us that we also have the opportunity for a tremendous biomarker, very much building on Martina's brilliant work and the work of many others who have collaboratively worked for many years in this space. But the idea is that, with the treatment with the drug, with the tool compound that is sort of the inspiration for the future drug, we can actually detect an increase in uromodulin in the urine of these animals, which again suggests that we're restoring the kidney function in these animals. And we also have a readout for when we are doing that. So if we can measure something in the urine that tells us that the drug is working, this is a huge advantage for being able to take our program toward the clinic. And so I'm just showing you here some of the, we call this biomarker development that one can do for this disease. Silvana, who gave a talk at this summit last year from our group, presented some of these data, but I just want to remind everyone that she has done a brilliant job in this study in collaboration with Martina and Stan and many others. to actually take tubules from the kidneys of these animals and isolate them and then test the effects of the drug in these tubules. And as you can see, the red in the middle is where the uromodulin is not supposed to be. It's accumulating, aggregating inside these tubules in a way that is not good for the tubules. It's not functional. However, if you treat the tubules with the drug, you can see that the uromodulin becomes normal. This is what it's supposed to look like, to have this linear pattern on the surface of the tubules. And you see this also better here, which basically means, again, that we're able to restore the normal uromodulin where it needs to be, while hopefully getting rid, as you can see, of all the bad uromodulin that is accumulating inside the tubules. So very nice results here as well to show that the drug is working in this setting. And so, to summarize, essentially, we have developed an understanding, a much deeper understanding of this nodal target, the TMED cargo receptor target. And as I mentioned to you, we are now doing work in retinitis pigmentosa and a couple of other proteinopathies as well. I won't show you the data, but just to tell you that this is actually an advantage for ADTKD in the sense that you can actually group this disease together with others, because that can increase interest in this therapeutic strategy, and it can bring more attention to this program, if you will, and potentially more investment as we move forward, because it allows us to understand biology that is relevant to many more diseases than just ADTKD. So I want you to all see this as especially for the patient side, to see this not as us being unfocused, but actually being doggedly focused on finding a solution to this disease by taking this nodal approach that allows us to increase the applicability of this biology and this program to many other diseases. So it is an advantage to be able to take this nodal approach for this disease. Now, you could say, well, what if any of your efforts with the small molecule don't work? You just told us that there's a lot of opportunity for failure. And I'm, again, continually humbled by the complexities of drug development. So as an alternative, we're not sitting on our hands. As an alternative to the small molecule compound, we actually are taking advantage of a different technology, which is called antisense oligonucleotides, or ASOs. This would be an injection in the arm. It's otherwise known as a nucleic acid therapy. It wouldn't be a pill that you take, but it would be an injection in the arm. And just for a brief reminder for those who may not know what ASO technology looks like, I'm taking as an example the MUC1 mRNA. So this is a kidney cell. This is the nucleus of that cell. And here's the MUC1 mRNA. So the message that Martina so elegantly explained to you, from the DNA, we get to the message RNA. And this is the ASO drug. So it's a small sequence that is complementary to the mRNA. And then we provide it to the cells. The cells take it in, the kidney cells take it in, and the ASO goes into the nucleus of the cell, and it finds its complementary mRNA, binds it. And that binding event, it turns out, when you have a good ASO, results in the degradation of the mRNA. So we are preventing the mutant protein from being produced. The protein is not even made because we are getting rid of the mRNA from MUC1 in this example. One can do this for any other gene. And so the idea is that we're able to develop a completely different approach, unrelated to the biology that I previously shared with you with the TMED cargo receptors. It's a completely different approach that allows us to essentially get rid of the mutant mRNA in this case, right? So this is a different approach to this. And I'm just showing you that we have actually been able to successfully develop an ASO. In this case, I'm showing you an ASO for MUC1, but we're working hard on additional approaches for UMOD as well. And this is an ASO that was given for 3 months, for 6 weeks, actually, to this mouse. And we have not seen any toxicity, but we have seen more than a 75% reduction in the mutant protein as a result of using this ASO. So we have a very impressive reduction of the MUC1 in the kidneys of these mice. Again, we're only curing mice so far, but we're trying to get there for pushing this project forward toward humans as well. And one of the key points here is that there's no toxicity, because there's been a lot of concern that these ASOs may confer toxicity. And so we are not seeing any of these signs here, which is a very, very encouraging signal. So what this means is that we now have another shot on goal for this disease using this technology. And of course, we're exploring all of the nucleic acid technologies that we might be able to use to treat these diseases. And so I'm just giving you this as an example to show you what we're doing. And so if I were to summarize today where we stand, we are very much pursuing our TMED targeting compound, the pill, if you will. Ultimately, it would be an oral pill that we can take for these diseases, which would be both for MUC1 and UMOD. And we're also pursuing early days, but we're pursuing ASOs and other nucleic acid therapies that would be delivered as an injection in this case. But those would also be therapies that can really, hopefully, as we see in the mouse models, adequately address this disease. And so I just want to end by where I began and to basically say that we really do live in an era where we have more tools at our disposal than ever before. I am very determined that we will do everything we can to get there in terms of the ultimate goal at the summit. I am humbled by the complexity and the difficulty of the path, but not daunted. I think that we have every opportunity to continue to fight to get up to the top. And I have no doubt that together, working together, and I think this is really important as well, that it takes a lot of stakeholders working together, including the patients and the families, all of us together to be able to reach our ultimate goal. And so, on behalf of a very grateful team at the Broad Institute, I want to thank all of you for your attention today and for this opportunity to share with you some of our recent progress. As you can see, it also takes a village to get all this work done. And I'm very lucky to have an incredible team of scientists working with me. And also an incredible team of collaborators. You've heard from Martina. We work so closely with Tony, with Stan, Kendra, and Greg also in Cyprus. And, we couldn't be luckier for all the other collaborators. You heard about Peter Conlon and many other investigators all over the world who are working together with us in every country, really, to be able to build, the registry, the understanding of the disease, and ultimately the therapies that will allow us to get there. And so I encourage all of you to always stay in touch with us. We are always inspired by collaborating with patients. And again, I thank you very, very much for this invitation, and we'll be happy to take any questions for any time that we might have. Thank you so much.
Thank you so much, Anna. I'm just sitting here. I have goosebumps. I'm just so excited to hear where you're going, the efforts, and your commitment, your undying commitment and optimism is it's making my day and my life because I think of what this means for my sons who have yet to be tested, but odds are at least one of them has this. And I don't want them to experience what my parents, my families have. And I'm speechless. So just thank you so much for being here and being such a valued partner in all of this.
Thank you. That's wonderful.
Let me turn it over to Lisa and see. Let's see, we've got about 10 minutes for questions. What
good.
questions do we have for Anna?
Good morning, Dr. Greca. Let's see here. Okay, we've got a couple of questions. First question is, earlier publication claimed BRD 4780 to be the chosen compound for MUC1 dependent ADTKD. What was the reason for testing BRD 7635 for MUC1 when TMED is central protein for both MUC1 and UMOD?
Yes, all of these are tool compounds. So they're all, 7635 is an improved version of 4780. And we continue to make improvements as we understand more and more about the target. As I mentioned, now we are structurally enabled to some degree. This is an AlphaFold structure, but it's based on the cryo-EM structure that we published a couple of years ago for TMED9 homomeric structures. And so, as we understand more about the target, we can build better and better compounds. So 4780 and 7635, I want everyone to understand at this moment, they are tool compounds. They will never become drugs that go into humans. They are just tools that we are using to understand the disease. We initially thought that they might have legs to go into the clinic, but that has since proven not to be true. But we are now on the road of building better and better small molecules that will eventually, one of them will be selected to go to the clinic. And we're closer than ever to that. But it's a process. And, this is what I mean that like when you're climbing up the mountain, sometimes you think you're getting somewhere, but then you have to stop and backtrack because, you discover that there's a toxicity or a liability with some of these compounds. And then you have to design new compounds where you're selecting away from that liability and more towards specificity for the target that you're going for. So I hope that this helps answer the question, but we're getting smarter and smarter about developing an ideal small molecule that can hit this target. And these compounds are just on our way to get there, essentially, right? Like the iterative process of getting there.
Wonderful. Okay, next question. CRISPR research for ADTKD. Any update?
Yes. So we are also working on CRISPR technologies for those who are familiar, this would be opportunities to actually cure the disease. So it would be a one and done approach using CRISPR technology or other such molecular machines like CRISPR, for example, base editors or prime editors that were discovered by my colleague David Liu at the Broad. We are very much working on these approaches for both MUC1 and UMOD. However, there are many hurdles to get there because nobody right now knows how to efficiently deliver these kinds of molecular machines into the proper cells in the kidney. So we are continuing to do a lot of discovery work in that area in collaboration with the Liu Lab at the Broad, who are the world's pioneers. So for anyone who saw, the best, perhaps the best example, the most beautiful example of what CRISPR can do is baby KJ, who was in the news last year, this little baby in Pennsylvania who was treated with a base editor in the liver. So the delivery to the liver is solved, and it's something where we can actually much more readily deliver these tools. And yet it's even early days for that. So the kidney is behind because we haven't really solved the delivery problem. And so we have much work to do, but we are doing it. So I want to make sure that you understand that that's an approach that will take much longer, but we are very much determined to work on that as well until we can solve it. So very much ongoing work. I didn't speak about it today because there's not much to say in terms of updates. It's all sort of boring technical details that we're working on, but we are working on it, very much so.
Okay, we've got one more question here. I'm guessing this falls under the heading of things that are humbling about the drug development experience. But can you speak a bit about whether funding is or has been a challenge along the way, especially given these diseases are rare?
Mm-hmm. Yes, absolutely. I mean, there's always this issue of, this is part of the reason why we started the accelerator at the Broad, because it is often the case that these diseases are relatively neglected in the drug development process. Again, especially since I mentioned to you how perilous the process is. So if the patient population is small, you can imagine that doesn't motivate a lot of people to work on this problem. However, genetic diseases, also rare genetic diseases, have their advantages. In this case, we can easily identify the patients using genetics, which is why it becomes of paramount importance that this organization exists and that this summit exists every year. We need to raise awareness so we can find more and more patients so that when we are ready for clinical trials, we have a patient population that is ready. This is very attractive to those who make investments into this space. So the companies that build clinical trials for rare diseases want to see a patient population and clinicians who work with these patients so that they can execute on these clinical trials as efficiently as possible. So this foundation that we're all building here together for the next 2 days is crucial for being able to get these new potential therapies over and into the clinic. And so everybody needs to come together, the clinicians, the scientists, the patients, the families, the foundation, the regulators. It really takes a village. All the investigators have to work together so that we can build the necessary foundation for clinical trials to be executed as efficiently and as quickly as possible. So funding is always a challenge. I'm constantly fundraising for rare genetic diseases, including ADTKD. We have been blessed with funding so far for all of our different scientific projects. And we want to make the best of this truly a gift that is bestowed on us, the gift of funding, to make the best of this resource so that we can actually get as many shots on goal as possible and build as many therapies as possible that we can advance toward the clinic. So, and go as fast as we can, of course, always maintaining, the protocols and safety and all these things that need to happen in order for treatments to make it from mice in the lab to patients ultimately. So thank you for your question. It's an ongoing struggle, but one that I welcome because I have a great sense of sort of purpose and meaning from this work. And I think I speak for our entire team when I say that we feel a great sense of purpose in doing this work on the benefit of patients everywhere.
Thank you, Anna. We have couple more minutes, and I'd like to just ask you to take a step back. Where do you see this field in, say, 3 to 5 years, 10 to 15 years? I mean, understanding that, you can't predict and it's a
Yeah.
mountain and ups and downs, but give us a vision for at least what you think is feasible and what you're working on.
Sure. I mean, yes, it's always difficult to make predictions, of course, and as I mentioned throughout my talk, I've been humbled many times, and so there's always the possibility that we try to go up the trail and then we have to backtrack and regroup. But I think over the next 3 to 5 years, I predict that we will have one or two clinical trials starting in this space. I think that's a very realistic goal. I think that we have every reason to believe that the small molecule program and potentially the ASO program will make it toward the clinic. I'm very bullish on that. I think that we have a good chance to at least take our best shot, right? Like that's all we can ever do, and hope for the best. And I do think that over the next decade, we are going to make substantial progress toward the gene therapy goal, because ultimately what I'd like to see maybe toward the end of the arc of my career, I would love to be able to cure these diseases, right? So this is not something that we will be able to do in the next 3 to 5 years. I think that will be for the kinds of treatments that manage the disease, like the pills, the ASOs, these kinds of things. But then for a cure using CRISPR, base editors, prime editors, these kinds of technologies, maybe the 10 to 15 year horizon seems more realistic. But I think that that's also very hopeful. And I speak of that as a physician, as a scientist, and also as a mother myself, right? I mean, I would love for these treatments to be available so that the next generation doesn't have to worry about, what our colleagues and patient friends mentioned earlier. Cliff, for example, this worry and stress about sort of managing chronic genetic disease. Hopefully, the next generation wouldn't have to suffer from that. But we have a lot of work to do in order to be able to do that. So, and again, I may be humbled, and please, nobody here hold me to the timelines. I know this is recorded, and you can bring it back in a few years and say, Anna, you said, all I can say is that everybody here will do the best we all can, right, to meet those timelines or even accelerate them as much as possible. But we do need to reach the clinic over the next, certainly 3 to 5 years is, yeah, I think that's a realistic goal. And then for developing, the kinds of tools that would lead us to a cure, I think that's a longer horizon, a 10 to 15 year horizon.
Thank you so much. That's helpful. And we promise not to hold you to those numbers, but to
Oh,
just
thank
to
you.
give people a sense of time in all of
Right.
this, given the
Yeah.
complexity it takes.
Yeah, I mean, this is what I want to convey to everyone. The fact that I'm not, I'm saying, don't hold me to the time is not because of any lack of determination or commitment, but it's because with all of that, you can get to an unpredictable sort of point where you have to backtrack and then you're delayed. And we've already seen some of this happen in these programs, but that doesn't deter us from continuing to move forward, learning, regrouping, and being able to bring the project forward. And so ultimately, this is what it takes. If you look at any other, for example, I invite everyone here, I'm sure many of the scientists know this. If you look at the revolution that happened with cystic fibrosis, the patients, the foundation, and the scientists, the physician scientists, worked for years in order to be able to bring these therapies that now make that disease, I mean, they have totally revolutionized the outcome for that disease, right? And so these patients, these children with cystic fibrosis are now, living basically a normal, life, right, with those treatments. But what did it take to get there? There were so many twists and turns along the way. And, if you ask seasoned drug developers, they will tell you that every drug development program dies at least 3 times before it's actually resurrected to make it all the way to the finish line. And so we just have to be determined and, continue to fight and move forward, understanding that, there's twists and turns along the path. So as long as we're all united in that understanding and we work together, I have no doubt that we will ultimately get there. I mean, our being together today is a testament to that. But at the same time, we have to be, hardworking, realistic, resilient to take the setbacks and then continue to move forward with determination. So that's my message for today.
Which is greatly appreciated. And your team's commitment and persistence is just palpable. And so we just are so grateful for that. So thank you for joining us
Thank
today.
you so
I know
much.
you need to take off, and we'll be in touch. Best to you.
Thank you. Thank you. Greetings to all.
Okay. Next we have, where are we here? There we are. Maria Pia Carteret. I hope I said that correctly. She has a master's in molecular and cell biology from the University of Bologna and a PhD in molecular medicine at Vite Salute San Raffaele, University of Milan, in the molecular genetics and of renal disorders unit led by Professor Luca Rampoli, where she is currently a postdoctoral fellow. Today, Maria Pia will be presenting cutting-edge research that I first became aware of last year at last ASN, and was just published last month in the Journal of the American Society of Nephrology. And with that, I'll turn it over to you.
Hello, I hope you can see my presentation. Thank you very much, Sharon, for the kind introduction. Thank you for the invitation to share with you our latest results on the effect of calorie restriction on ADTKD mode progression. In the lab, we are interested in the molecular genetics of renal disorders, and one of the focuses is ADTKD caused by mutation in the UMOD gene that was already nicely introduced by the previous speaker. I would just like to mention that all the mutations reported so far are missense, and they all are predicted to impact on protein folding. And evidences in patient kidney biopsies and in different in vitro and in vivo model of the disease demonstrated that mutant uromodulin is retained, so it's blocked, in the endoplasmic reticulum ER of expressing cells. In the lab, we generated a transgenic mouse model recapitulating all the features of the human disease. And several years of work allowed us to reconstruct the natural history of the disease. We observed that mutant uromodulin is ER retained already at 1 day postnatal in kidney of mutant mice. Starting from 1 week of age, we observed progressive accumulation of mutant uromodulin. We demonstrated that inflammation is an early event of pathogenesis that already occurs at 1 week of age. And starting at 12 weeks, mutant mice start to develop urinary concentration ability defects and progressive renal injury, leading to mild kidney failure at 24 weeks. Now, in this slide, I try to sum up all the knowledge about the ADTKD mode molecular pathogenesis, so all the pathways that are demonstrated to be induced by mutant uromodulin expression. Different studies reported the induction of ER stress and the unfolded protein response. Other defects are regarding the mitochondria or increased cellular death of those cells. But today I would like to focus on this phenotype that is autophagy impairment, meaning the block of the degradative mechanism of the cells, that was described in different mouse model of the disease, and that in our model is demonstrated by the accumulation of P62, specifically in cells expressing mutant uromodulin. Now, P62 is a specific protein that mediates autophagy, so it is recognized by the autophagosome and degraded along with the substrate. So, the accumulation of this protein in mutant uromodulin expressing cells is a marker of impaired autophagy in those cells. We also reported that in kidney of mutant mice, phosphorylated level of this protein, that is S6 ribosomal protein, and that is a known downstream target of mTOR, the master regulator of the cell metabolism, is strongly induced, again specifically mutant uromodulin expressing cells, suggesting that this could be upstream to the autophagic impairment observed. Now, based on all these knowledges and trying to integrate an anti-inflammatory strategy with an autophagy-inducing approach, we decided to test the effect of calorie restriction in the context of ADTKD human. This is a dietary intervention that, by acting of a plethora of molecular mechanisms in the cells, has been demonstrated to be beneficial in different settings, characterized by toxic accumulation of toxic protein and chronic inflammation. So experimentally, we used transgenic mice expressing both the wild type or the mutant uromodulin, and those mice were fed ad libitum or with a 30% reduction in calorie intakes, starting from 8 weeks, that in mutant mice corresponded to an early stage of the disease, characterized by sustained inflammation and fibrosis, but still normal kidney function, until 23 weeks of age. We test the effect of this dietary intervention on mutant uromodulin, and we observe that we observe a strong reduction in mutant uromodulin near retention, as demonstrated by reduced colocalization of the transgenic protein with the R marker calreticulin, that correlates with a strong reduction of mutant uromodulin aggregates, demonstrated by the reduction of this high molecular weight smear that you can see in this Western blot, and that is likely corresponding to these huge intracellular perinuclear aggregates that, as you can see, almost disappear after diet. Next, we study lipocalin-2, that is a well-known marker of distal tubule damage, that, as you can see, is strongly induced and again specifically observed in mutant uromodulin expressing cells, and that after diet is dramatically reduced along with the mutant uromodulin aggregates, suggesting that reducing the accumulation of mutant uromodulin can relieve the stress of those cells. And a reduction in lipocalin-2 levels is also observed when we test urine of mutant mice after the diet. Through histological analysis, we also demonstrated an amelioration in tubular dilation, in inflammation, and also in interstitial fibrosis in kidney of mutant mice upon calorie restriction. Now, to assess the effect of the treatment on disease progression, we decided to compare 23 weeks old mutant mice after 15 weeks of controlled diet with 8 weeks old mutant mice, corresponding to the age of the mice at the beginning of dietary intervention. As you can see, ER retention of mutant uromodulin is already present at 8 weeks. It progressively increases at 23 weeks when mice are ad libitum fed, so suggesting disease progression. But in kidneys of mutant mice after calorie restriction, we observe levels of mutant uromodulin near retention that are even lower to those measured at the beginning of dietary intervention, suggesting that this dietary intervention not only prevents the progressive accumulation of mutant uromodulin, but it also clears protein aggregates that are already present. We also studied the kidney function of our mice, and we observe a progressive rise, so a progressive increase of blood urea nitrogen and diuresis levels in ad libitum fed mutant mice, while levels of both these two parameters remain stable and within the normal range when mice underwent the diet. And we also observed that parameter of kidney damage, so tubular dilation, inflammatory cell infiltration, and interstitial fibrosis, at the end of the calorie restriction, show levels that are significantly reduced compared to the starting time point, suggesting that initiating calorie restriction at early disease stage could, at least in part, revert the already established phenotype. And stemming from this observation, we wonder if calorie restriction could also be effective at an advanced disease stage. For this, we performed a second experiment, feeding mutant mice with the 30% calorie restriction, this time for 24 weeks, and starting at 24 weeks of age, so the age of the mice at the end of the previous experiment. As you can see, at 1 year of age, so at the end of the treatment, kidney of mutant mice show a very spongious aspect and a reduction in the kidney weight. But if we look at kidney of mutant mice after calorie restriction, both the morphology of the kidney and the weight are rescued. Regarding kidney function, again, we test blood urea nitrogen and diuresis levels. We observe a strong increase of both these parameters indicative of disease progression. But again, both the parameters were maintained stable after calorie restriction, suggesting that also at advanced disease stage, the dietary intervention can block disease progression. And similar results were obtained when testing again tubular dilation, inflammatory cell infiltration, and interstitial fibrosis. All these parameters progressively increase with disease progression, but are maintained stable by calorie restriction. And we also decided to test senescence-associated beta-galactosidase, that is a marker that was recently reported as a predictor of renal outcome in patients with chronic kidney disease of different origin. As you can see, this marker is already increased in kidney of mutant mice at 24 weeks. It increases and it is maintained stable by calorie restriction, suggesting that this treatment can prevent the worsening of renal damage. Now, we tried to have a more comprehensive view of the effect of calorie restriction, both at early and late disease stages. So we integrated all the parameters tested, and we performed this analysis that is called unsupervised K-mean clustering analysis. As you can see, we observed that all the wild-type mice form a single cluster that is clearly separated from the cluster formed by 8 weeks old ad libitum mutant mice, that again is very well separated from the 23 weeks old 23 weeks old ad libitum mice, underlining this disease progression. But if you look, the 23 weeks old calorie restricted mutant mice form a third cluster, well separated from the previous two, that is closer to the wild type, resembling the effect that we see experimentally on the reversal of the already established kidney damage. For the second experiment, we again observed a single cluster formed from the wild type mice, and the formation of two distinct clusters by the 48 weeks old ad libitum and the 24 weeks ad libitum mutant mice, while the 48 weeks old calorie-restricted mutant mice clustered together with their 24-week-old counterpart, so underlining their very close similarity and confirming the effect of calorie restriction in blocking disease progression. Going a bit more in the molecular mechanism, we also demonstrated that the effect that we observed on the protein, so on the mutant uromodulin protein, is not reflected by any effect on the transcript level, nor by increased secretion of mutant uromodulin in the urine, suggesting degradation as a leading mechanism. So we studied autophagy in kidneys of our calorie-restricted mice, and we observed that the autophagy the marker of impaired autophagy that I showed you before, so P62, is strongly reduced in kidney of mutant mice after calorie restriction. And we also observed a strong reduction of phosphorylated S6 ribosomal protein, so the mTOR downstream target, suggesting that calorie restriction quench mTOR hyperactivity in mutant uromodulin expressing cells, hence restoring the autophagy impairment in those cells. And additional molecular insight derives from the study of the autophagy, that is the selective autophagy of the endoplasmic reticulum, so the place in the cells where mutant uromodulin is retained, and that is mediated by specific proteins or autophagy receptors. And we observed that genes encoding for some of those receptors are significantly downregulated in kidney of mutant mice compared to the wild type, and they are restored to the basal level after calorie restriction. And among these proteins, I would like to mention just a specific isoform of FAM134B that is encoded by RETREG1, and that was previously demonstrated to be transcriptionally induced by nutrient stress to mediate starvation-induced autophagy. So overall, this is the model of our work. So we demonstrated that calorie restriction reduces ER retention of mutant uromodulin, and this is likely downstream to the effect of calorie restriction in restoring autophagy and possibly ERphagy in mutant uromodulin expressing cells, with a concomitant degradation of mutant uromodulin. This can, in some way, relieve the overload on the ER machinery, relieving cell stress, and this effect, together with the direct anti-inflammatory effect of calorie restriction, quench fibrosis and prevent the induction quench inflammation, and prevent the induction of inflammation and block kidney function decline. I would like to finish by thanking all the members of the Molecular Genetics of Renal Disorders unit, all our internal and external collaborators of this project, our founding agency, and all of you for your attention. Thank you.
Thank you so much, Maria Barra. That was just fascinating. I'm really excited to hear questions that folks might have about this potential treatment. I know this is in the very early stages of research, but yeah. Lisa, do we have any questions?
Hey, yes, good morning. We do have questions. First question is, per calorie restriction discussion, does the research support or suggest a sort of fasting or periodic calorie reduction to slow disease progression? Surely consuming 70% of daily caloric need would be unsustainable long term.
This is, I think, the greatest limitation of this work. I also think that it's not easy to compare the study that we perform on mice with the direct translation in human, because we cannot directly compare the mouse metabolism with the human metabolism. So for sure, further study will be needed to dissect also the molecular mechanism that we induce with calorie restriction. Also, thinking to, I don't know, employing different strategy such as mimetics of the calorie restriction, so pharmacological compound that can activate the same pathways that we activate with the calorie restriction in the mice. But for sure, further study will be needed. This is really the first step of a very long path, I think.
Well, there's another question. It says, does the calorie restriction research translate to Mach-1 variation?
This is a very interesting one. So, in principle, it could work also in the MUC1. Of course, we don't have data by now, but we will be really interesting to test it also in the MUC1 model. In principle, it could be effective because MUC1 should be degraded through the same degradative pathway as uromodulin, at least at the end. So maybe we can act also on the accumulation of mucin 1 by similar strategies. But we will be very interesting to test.
Another question. What happens if you stimulate I'm probably not going to say this right, autography with urolytine? Urolitine.
Sorry, can you repeat the?
So it says, what happens if you stimulate autography with urolytine? Urolitine.
Yes, I get it. I don't know the answer. We don't never think to this molecule, but I think that potentially, and actually there are data in cellular model showing that also the pharmacological stimulation of the autophagy acting on mTOR, so the master regulator that I was mentioning, could be efficacy in inducing autophagy and so degrading the protein. So I think that also pharmacological stimulation of autophagy could be effective.
Wonderful, thank you. Next question, would a GLP-1 drug be helpful to facilitate calorie restriction?
This is another interesting one. We are discussing in the lab actually in this on this class of compound as a part of the calorie restriction mimetics that I was mentioning. It's not clear actually if GLP-1 activates autophagy. There are still scientific debate on this. Potentially it is interesting also because they are now fashion drugs. But I don't know. It needs to be tested firstly on the mouse model to see if we converge on the same molecular mechanism and we can have the same result. We can mimic the same result as the diet.
Well, thank you. Has the possibility that certain foods need to be restricted in lieu of total calorie reduction been explored?
This is another path that can be explored. We decided to test this type of protocol, so we horizontally restrict the calories. We don't know if there is a specific macronutrient that is contributing to the main effect that we see. for sure the effect that we see with calorie restriction is really pleiotropic, so we are inducing different pathway. So probably restricting just one specific kind of food, its efficacy in activate one pathway, but not, for example, to have the anti-inflammatory effect of calorie restriction. So these are all experiments that we are really interested to do. Also to dissect the diet, so restricting one by one all the macronutrients, so proteins, carbohydrates, or lipids, to see if we have a specific macronutrient driving the main effects.
Well, that's all the time we have for questions today, and I'm just grateful for your being here. It sounds like you've got a lot on your plate in terms of future research, a lot more to learn about this.
Thank
So
you.
thank you for joining us today.
Thank you very much for the invitation.
Okay. Whoops. We're going to take a break. There we go. Coming up next after the break, Dr. Andrew Lazar. But right now, we will take about a 10-minute break. Let's say we'll see you back here straight up around noon, minute or so after. Um, and uh, and yes, thank you for your attention so far. We look forward to seeing you again in 10 minutes. Welcome back, everyone. Okay. So this morning's sessions, we heard a lot about cutting edge research on ADTKD and cutting edge research on therapeutic advancements. But we can't learn about ADTKD if patients aren't getting diagnosed. So this afternoon, we're going to hear from 3 experts on how to integrate genetic testing into clinical practices. We're first going to hear from Dr. Andrew Lazar, the Chief of Nephrology at University Hospital's Ahuja Medical Center and Clinical Trials Director at University Hospital's Cleveland Medical Center, who will present a case study and share his clinical insights on when and how to refer patients for genetic testing. Next, we'll hear from Maggie Westmeyer, a genetic counselor and Director of Clinical Genetic Services in Organ Health at Natera, who will discuss the support provided to nephrologists before, during, and after referring a patient for genetic testing. And then finally, we'll hear from Dr. Madhvil Lapa Rupali, a nephrologist affiliated with Memorial Hermann Health System and Houston Medical Hospital, who will describe how she has seamlessly integrated genetic testing into a 15-minute clinical visit. And you should each feel free to share a little bit about yourselves, your background, if you like. And we'll hold questions till the end of the entire panel presentations. All right, let me turn it over to you, Andrew.
Okay, I really appreciate the opportunity to be here. You can hear me okay, Sharon?
Yes.
Great, great, great. Thank you so much for having me. I am I'm the head of clinical trials, University Hospitals of Cleveland, focusing mostly lupus, IgA nephropathy, APOL1. But I'm really a city-dwelling nephrologist. I just see people every week. And I really wanted to share my journey of going through that revelation of someone who didn't really appreciate the impact of genetic testing to going through this journey and now recognizing that, wow, in 2026, when I see people, 50 to 100 people outpatient every week, that we have to fight to make a diagnosis. That when we leave people undiagnosed, we have trials and we have drugs that are coming, and we have to get people in trials, and we must make a diagnosis for the patient's sake, and they are counting on it. So I just wanted to share, this is the kind of patient who I would see a number of times per week. Mr. X, 45-year-old man. Today, between Google and ChatGPT, people come to the office and they already know what they have, right? This gentleman comes, he has been told maybe he does have ADPKD, he's got bilateral kidney cysts. There is a significant family history of PKD. Many of his family members have CKD and they have kidney cysts. Yes, the GFR's dropping, but the cysts are stable. The kidney size by ultrasound is only 10.5 centimeters bilaterally. So you look at this kind of case, so many of the cases that I see every day, all week long, I can't say with certainty what someone has. And yet, I think that we have to fight to understand what people have if we want to tell people what your prognosis is. Do we have treatment options? So why would I think about genetic testing here? What does Mr. X suffer with? Kidneys are not particularly large. That doesn't mean he doesn't have PKD. We order a CT. We're waiting on total kidney volume. Now, if this is PKD with a somewhat atypical course, I'd like to know what this gene is, right? Now, if they have a truncating PKD1, that is worse. A non-truncating PKD1, maybe ever so slightly better. PKD2, better yet. But there are a number of other genes now, IFT140 and others, for which I'd like to know what that gene is. So now this patient, this could be an atypical PKD. It could be a different disease. This is now something that I would really still call CKD unknown etiology. This is really this is a class of disease now in nephrology, CKDU, CKD unknown etiology. And this is what I think about every single day. Why do people come to see me? They want to know, what do I have? Why? What will happen to me? What are the treatment options? And I want all of my colleagues to really think through when we see a patient, this is their anxiety. And then they also want to know, will this affect children? Will this affect nieces, nephews, siblings? So we have a lot of data out here published that, and this just happens to be one in the German CKD study, where they looked at over 5,000 patients just who had GFRs of 30 to 60 or quote unquote overt proteinuria. That's all they looked at, and they had a group of people who went through every single chart to say, do we know what this patient has? And they felt when all was said and done that 20% or more were really CKD unknown. They didn't really know. Now, I would argue that we way overdiagnose diabetics as having nephropathy related to that diabetes. Should someone have hypertension, this is probably not the cause for their CKD. You have to look further. So when I started seeing these data and I looked at my clinic, I really felt that it's probably upwards of 40% of my patients that I don't really know. And that's not good enough today. Now, you start looking at what's the impact of doing broad genetic testing for this group of patients who have CKD unknown, and trials like this, where they looked at a group of over 440 patients, had a diagnostic rate of 22%. Now, there's something about this 20% number when you look at all sorts of recent trials of doing broad genetic testing for those who may have a family history, they have unknown etiology. All of these trials are showing somewhere in this 20 to 22% range. There is no other test that I order to rule out disease X, Y, or Z, doing an echocardiogram, doing an SPEP, UPEP, looking at different antibodies, that I have a diagnostic rate of 22% that changes what I do for that patient. So we've got to think about the integration of genetic testing for many, or even most patients who we see in CKD clinic. This is yet another one out of Columbia. They looked in their CKD clinic and said, if you meet just one of these three criteria, you have a family history of CKD, a suspicion of genetic CKD, which they didn't define further, or you have CKD unknown etiology, I'm going to do a broad genetic test. And they found 92 patients who they did a broad genetic test. And for those 92, 22 of 92, 24%, they found an answer for why that patient had CKD. They had X-linked as well as autosomal output, dent disease, HNF1B. Here is one of the ADTKDs, UMOD, TRPC6 FSGS. That's something I want to know. You know, a TRPC6 patient may respond very well to calcineurin inhibitors. Little syndrome. Do I want to miss a Fabry's when I have Fabrazyme so I can treat this patient? So these are not obscure diseases. These are diseases that either have treatments or we have ongoing trials. So we have to fight to make a diagnosis. And not to mention the fact that we have guidelines now. This is from the National Kidney Foundation guidelines from just December of 24. You see here on the left, this box. These are specific recommendations for whom we ought to consider genetic testing. Do they have a family history? Anytime you're thinking about a biopsy, you need to add genetic testing data that will really enhance our ability to interpret that biopsy. CKD, unknown etiology, cysts. We have to think about adding on genetic testing. We went ahead and we do a renacite, in this case, and boom, this was not PKD, this was not IFT 140, UMOD. So this now. We want to know broadly when we have CKD, knowing that I have a positive genetic test is very important prognostically. When you look at this particular trial of over 5,700 patients, CKD of any cause, having a positive genetic study, we know that they had a 72% higher risk of kidney failure. So going back to what do I have? Why? What will happen to me? And what are the treatment options? I can't answer these questions without doing everything I can today to make a diagnosis. And for my CKD unknown etiologies, which is probably upwards of 40% of the patients I see, I may have a yield, as high as 20%, if I go on and I do genetic testing. When you look across MUC1, UMOD, HNF1B, these are tough cases that go undiagnosed. The sediment is bland. They have minimal proteinuria. You see the different genes below here. HNF1B, we may have certain clues, hypomagnesemia, they get diabetes, gout at a young age, UMOD especially, at least 50% have gout before about 45 years old, but they don't have metabolic syndrome. So maybe we ought to think about doing genetic testing for those patients because these may account for upwards of 2% of people who are sitting on dialysis undiagnosed. So really, again, this case, they want answers. I couldn't have given them the answer without employing genetic testing. So in the last couple of slides, I just wanted to include this. When you look at studies like the Irish Kidney Gene Project, positive family history, CKD, CK uncertain etiology, extrarenal features, they have broad genetic testing. Yields were about 50%. And look at MUC1, way, way up here. And MUC1 is not easy to diagnose with next generation sequencing. We have to go a little bit further. And look at UMOD. Two of the top six in this trial. So ADTKD is probably a much more significant part of everyone's practice. They just don't know it. Now, we have this gene problem. We have a disulfide bond problem. We have misfolding of this protein that is now, quote unquote, stuck in the endoplasmic reticulum, causing interstitial fibrosis. Now, when you look here to the right, you can see those clumps of red. You are seeing that red UMOD is now inside cells rather than here on the left, where you have a ribbon on the apical tubular membrane where it should be. When it's clumped there within cells, that's what turns on an inflammatory cascade and leads to that tubulointerstitial fibrosis. Now, I just highlight BRD 4780, Dr. Brecker's group, that now this drug looked to have some off-target toxicity. So unfortunately, this may not be the drug that we see in human trials soon, but this is to make the point that we have very, very bright people, many who are on this call, as I have read a lot of your literature, that you look here, the upper right, that green clumping of MUC1 within cells causing interstitial fibrosis with drug exposure below, you no longer see it. It has been redirected to lysozymes for degradation. So, and you may not see it too well here, but the top you see purple with CD45 inflammatory cells. The middle left here with drug exposure, animal studies, of course, you don't see the CD45 inflammation. The right upper, you see fibrosis. The middle one to the right, you don't see that fibrosis. So understand that in clinic, all of my colleagues please that the research bench is like right next door to us. It's very adjacent. We have to make a diagnosis and get people to these trials so that anyone who I see who has family history, early onset CKD unknown etiology, I order genetic testing. 3 to 4 weeks later, I can get an answer and I have access to a genetic counselor. So this is how we need to start to incorporate to fight to make that diagnosis. So I appreciate it. I hope I didn't go beyond my little part. I can't hear you, Sharon.
Sharon, I think you're muted.
I am. Thank you. That was excellent, Andrew. Thank you so much. And we'll move now to Maggie.
Sounds good. Thank you. So it's nice to speak with everyone today. I'm a genetic counselor and have been with the laboratory Natera for 14 years, working in renal genetics for the past almost 6 years, which has been really exciting. And I work with a great team of genetic counselors who speak with patients every day about their test results. We also speak with clinicians to help clinically correlate and understand genetic test results. And have made a number of ADTKD diagnoses through our Renasight test. So today I'd like to just discuss the role of the genetic counselor, and after a positive test result and why genetic counseling matters in ADTKD. So we'll talk about how genetic counseling can help reduce diagnostic uncertainty, identify at-risk relatives, prevent unnecessary procedures, inform kidney donor decision-making, support reproductive autonomy and decision-making, address psychosocial burden, and how this bridges the molecular genetic testing and clinical nephrology. So genetic counseling really translates molecular findings into informed medical and personal decision-making, by translating information that can be complex and difficult to understand into patient-friendly language. So when we receive a test result, this can really help clarify the diagnosis. And during result discussion, what the genetic counselor does is review the specific gene variant identified. In this case, in a UMA diagnosis, for example, we would review the inheritance pattern, so autosomal dominant inheritance. And we would help correlate the genotype with the patient's phenotype. So help the patient understand the clinical features that can be expected with the condition diagnosed, including progressive CKD, the findings of bland urinary sediment, hyperuricemia or gout that's often early onset, and understand that there can be variable age at onset. And so which can lead to intrafamilial variability. So not everybody in the same family, even with the same condition or genetic cause, can have the exact same symptoms or the exact same age at which they have kidney disease. So that's something that we would review. And in some cases, a patient may have testing before having symptoms, and we can discuss the limitations of genotype phenotype prediction, meaning what's the exact signs and symptoms that someone's going to develop based on a positive genetic test result. So to discuss what's known about that those genotype phenotype correlations, and in some diseases they're very consistent and predictable, and in others there's much more variability. I think a really important part of genetic counseling that is something we have training in and devote our time to is helping patients process the emotional impact. I think as we heard earlier today through some of our patients' stories, is the diagnostic odyssey, the length of time at which many families, particularly in ADTKD, how long they go without a diagnosis. And this can be years or decades, right? Where there's and also awareness that there's a disease in the family history. There's an awareness that this individual has a kidney disease, but all these years of not knowing what the cause is. So there can be a really profound emotional impact of just providing a diagnosis and giving a name to the condition. And, many families report these years or decades without explanation, having undergone multiple biopsies or misdiagnoses, have uncertainty around, whether there's something they did to cause this, self-blame for disease transmission to children, these sorts of things. So the genetic counselor really can help with validating emotional reactions, reframing the diagnosis, as an actionable finding, address guilt surrounding the inheritance, and facilitate family communication. So it's, I think, important for us to recognize that relief and grief can coexist at the time of diagnosis and help work through some of those feelings. So when we talk with patients about the inheritance and risk to family members, we would discuss autosomal dominant transmission and the 50% risk to first-degree relatives. We would explain that both males and females are equally at risk and can equally be affected with an autosomal dominant condition. And likewise, each pregnancy would carry an independent 50% risk to inherit a genetic alteration for an ADTKD gene, such as UMAD. So we would review specifically by taking a detailed family history, who in the family is at risk, and what the clinical screening recommendations would be for those individuals if they haven't had genetic testing. But discuss how identifying at-risk relatives enables, early nephrology surveillance, can help avoid unnecessary diagnostic procedures, and can really help inform donor eligibility when it comes time to consider a related family member for kidney donor, to be a kidney donor. So cascade testing is the phrase that we use in genetics for testing other family members once a molecular genetic diagnosis is identified in an individual. And this really is key because a negative genetic test result isn't as meaningful, unless the affected individual and the family or affected family members have a known molecular genetic diagnosis. So once we have that, we can then test specifically for that genetic variant in children, siblings, parents, and extended relatives, aunts, uncles, cousins. All family members who are at risk and are interested in testing could be tested for that specific variant. Most laboratories offer single site testing, which can also reduce the cost in some cases of the genetic test. And so the discussion points that we would review in cascade testing would be, the different issues with testing someone who's symptomatic versus asymptomatic. The risks of testing minors, generally we don't offer genetic testing to minors unless there's a medically actionable, meaning a specific treatment that would be available to that individual. Typically that's reserved for adults. And the living kidney donor considerations, and also discuss in genetic counseling, we always, address things related to insurance and discrimination protections. And so things like, perhaps establishing life insurance prior to having genetic testing as an option, and just understanding what the protections are that were afforded through the GINA Act in terms of prevention of discrimination in the workplace and in healthcare and health insurance. And also what is and isn't specifically protected under that legislation. Reproductive counseling can be applicable for individuals of reproductive age, or even those who are worried about their grandchildren and so forth, right? Their children's children. So family planning considerations. We would review, of course, the 50% risk of recurrence in each pregnancy, and we can discuss the various options. So I heard it mentioned earlier today that there's not prenatal testing for these conditions for ADTKD. That's not specifically the case and again, this is where it's key to have that genetic diagnosis. Once we have a genetic diagnosis in an individual, we can perform prenatal testing during pregnancy through either chorionic villus sampling or amniocentesis to test for that specific mutation that's present in the parent. So that is something that's available, is routinely offered, in the prenatal space. And so a patient would be referred generally, during pregnancy to a maternal fetal medicine specialist, and perhaps likely also have genetic counseling to help coordinate that sort of testing that's available during pregnancy. So the various options that someone who is at risk of passing on a dominant condition, right? The things that are discussed would be natural conception and perhaps postnatal testing or testing when an individual is an adult. And we talked about prenatal diagnosis. Another option for individuals is pre-implantation genetic testing for monogenetic disease, also known as PGTM, which is for couples who are can undergo in vitro fertilization, and embryos that are created through that process can be biopsied, and a custom assay can be designed to test for the specific mutation in the parent in the embryos. And then the idea is that only embryos that are unaffected and do not inherit that mutation, right, are transferred into the mom for conception. So that is a growing area, and there's more utilization of that over time. Our laboratory also offers that sort of testing as well. Use of donor gametes, so use of sperm donor or egg donor is also an option for couples who wish to prevent passage of an autosomal dominant condition to children or adoption. So I think it's important to recognize and just always remind everyone that genetic counseling is non-directive. We discuss options, we provide support and informed, values-based decision making. So we work with a couple and families to understand what their individual values are, what their desires are, and tailor our counseling to those. Another important part of genetic counseling is enabling family communication, so supporting disclosure within families. And so we can often provide a family letter that explains the diagnosis, the inheritance pattern, and testing availability, because the doctor that I go to is probably not the doctor perhaps that my sister goes to, or families live all over the country and are separated. So helping provides just a physical piece of information that describes what the genetic diagnosis is, what the mutation is, what the testing is that could be done for an at-risk family member, I think can be very useful. And we can help empower family members with guidance on how to approach conversations with family members, provide resources to their relatives, and also help patients navigate issues related to estranged relatives, general communication, generational communication differences, and cultural considerations. Practically, genetic counselors really help make testing available by assisting with ordering familial variant testing. We often end up seeing relatives of patients we've counseled. We can help coordinate getting kits delivered to at-risk family members, or communicating with someone else's nephrologist to help with clinic-based testing, interpretation of those follow-up test results. And we provide documentation of our counseling back to the nephrologist in the form of a letter, in order to make sure that there's that, continuity of care. So we really do act as a bridge between the laboratory, the nephrologist, and other medical providers, and as other family members. I think another important thing that genetic counselors make sure to focus on is connecting patients to community resources and support. So we provide referrals to the Rare Kidney Disease Foundation and other kidney disease advocacy organizations, research registries, clinical trials when applicable, and we provide reputable web-based resources. In addition, if there's an individual who is suspected of having something like ADTKD, but perhaps has a negative genetic test, but wasn't tested for the MUC1 gene, for example, that's not currently available really on any commercial panel in the US at this point, referring them to a research study to have that sort of testing. So that's something that we help with as well. So that completes my summary of genetic counseling services and the description, and I'm happy to answer any questions.
Thank you so much, Maggie. We'll hold all questions until Dr. Bhupali goes, and then we'll open it up. So let me turn it over to you, Dr. Bhupali.
Okay, let me share my. Okay. See, I think I'm muted. Okay. I hope everybody can listen to me.
Yes, we can hear you fine.
Okay, so anyway, my name is Madhavi Bhupali. I am a nephrologist in Houston area for the past 15 years. So I will tell my little bit of journey about why I believe genetic testing is very important and how we can do in this 15 minutes of the patient encounter, regular daily basis, how we can work in the clinic. So, this is a simple thing. I think everybody is, talking about the same thing. Why is genetic testing important? Why we want to do it. So I think there are a lot of, etiology that we don't know. Like if you actually know most of the ESRD patients, at least 20% of the patients actually are still no etiology. When we write this diagnosis on the form, sometimes we just say hypertension because we don't know the diagnosis actually, because never been seen any nephrologist, never had any workup done. So that's one reason that we need a genetic testing. So, and I'm actually doing in my dialysis patients also. And also, some of the diseases are runs in the family. So it's very important for them to have the genetic testing done, because when they get transplant, right, they also kind of affects it on the transplant. They know what kind of genetic testing, and especially if the family members wants to donate, especially with the polycystic kidney disease, they cannot really donate from the family. So it's very important to have that genetic testing to be done. And also people who are young at the, when they start in dialysis. So these are the few reasons. And if you go, this is the broad thing why we need to have a genetic testing done. As I think everybody mentioned, even Maggie also mentioned, family planning is very important when you're doing your, family, going about the genetic counseling, all those things, hereditary diseases, familial diseases, cystic diseases, young age. People who are diabetic, we all know that diabetes can cause kidney disease, but you'll be surprised people who are diabetes may have a different diagnosis, a different genetic disease that can cause kidney disease along with the diabetes. And there are so many other deficiencies because of the fact that there are different treatments. Not every kidney disease has the same treatment. We have a blanket treatment at the same time. People who has different genetic diseases has different strategies as well. So I'll tell a little bit of my journey when I started. It started in my fellowship back in Louisiana, where we used to see a lot of focal segmental FSGS patients because the population there is more African American. And at that point, we didn't have any genetic testing at that point. We just used to, do the biopsies if possible. If they're already end of the diagnosis, end of the, spectrum of the kidney disease, we may not even able to do that. So that's when I, found out and read about this APO1, gene that has been very, very notorious in African American. And, there was some trial just started and they're still going on, this amplitude trial. That's when I started really doing, research to see what I can do. When I started my practice in 2012 in Houston area. So I pretty much called every lab to see where they can do this testing. And at that point, it wasn't available until later, really about almost 5-6 years ago, the Natera rep came to my office and say, hey, you want to do genetic testing? I'm like, yes, this is what I've been really waiting to get that. So since that time, I've been doing a lot of tests from the Natera. So at that time, my focus was basically doing more for APOL, African Americans. So really doing that. So when I did that, this is my, basically had the results. If you see that, I have a pretty big range, a big percentage of APO1. At the same time, there's some other things also. If you look into that, that's when I started, it's not just the APO1. There's a lot more than APO1 that I found out that I want to really, look a little bit more, broader terms, not just the one area that I'm looking into that. So that's when I went to more, when I did a little bit more of that. This is my office people, results. If you look at that, it's still APO1 is my big one. But if you look at the other one, there are a lot of other things as well. PKD, it is, I think everybody, familiar, 20% of the population in my area has it. And if you see a lot of other genes that really don't see that, TTR is one of them, which is, I'm also, kind of focused on that because it is involved a systemic disease, heart, kidneys, nerves, a lot of things. So that's when I, started doing it. It's not instead of just started with APO1, but went on to having more diseases that I wanted to do that. These are the few examples that I do want to, give from my office. So this is a young patient, one of my nurses where I work at the hospital. She came out to me saying that I have this hematuria. It's not like she's not urinating blood, but every time they do a blood test, they see the blood, right? So of course she's been urology many times. They did a cystoscopy, multiple things. Everybody said, no problem. It's just a small thing. Don't worry about it. And multiple times she was treated as a UTI as well. So she came to me and said, and I asked her, she has no family history. She's from Romanian. She doesn't really have any family history that she knows of any kidney disease. So I did the test and of course it came back positive as output. So it's very important now that she needs to go back and talk to our family members, because sometimes it's so subtle things that we really don't know. So it's very important. So she was very happy that at least she has a diagnosis now that she can work on that. And she actually went on to trying for some clinical trials for the Alports as well. And she went and had her hearing test done. And surprisingly, she does have a mild hearing loss, which she did not know about it. So it's really, fascinating to see those things. The other one, this is the patient came from India, already has a diagnosis of advanced kidney disease, had a stroke because of the hypertension, and his renal ultrasound definitely showed big cysts, multiple cysts in the kidney, consistent with the polycystic kidney disease. Then people do argue, like, why do you need a genetic testing now, right? You have the diagnosis from the imaging, you have everything. But he never had any family history that he knows from India also. You have to realize some of the developing countries, they may not have any, family history because they've never been to any doctors. So it's very important. Other important thing is that why the genetic testing needed, because he's having a baby and he's all very worried about it. So we did the test and he has a PKD1, which he had liver and kidney disease. So other unfortunate surprise thing is baby, after my genetic testing, they went and actually had talked to the pediatrician, neonatologist, and they did this imaging, and the baby already had a cyst. So it's unfortunate, but at least at the same time, they came to know, it's overwhelming for them. He's on dialysis now, trying to get a, transplant, but it just helps the, family about the, kids when they're family planning and whatnot. So, and these are the same KDIGO guidelines. I think even Dr. Lazar also mentioned about it. When it came back, it mentioned the KDIGO also mentioned in the diagnosis of kidney disease, genetic testing is very important. And these are the recommendations they put, were the reasons that you would need to have a genetic testing done. So based on that, this is what I created for my office to easy for a regular visit. So every patient that comes to my office, we give this form. Right, we fill out the name and everything, and this is the questionnaire for the patient before the physician goes there, right? And it has I try to put as much layman terms as possible, basically telling that if the kidney disease started early, whether you had any kidney disease but not known any risk factors, and you had a cyst in the kidney or any protein, blood, had biopsy done, or I did put African American just because of the fact that I am, more biased on the APO1, that I'm looking for APO1. And surprisingly, some Hispanics also actually have APO1. That's why I put that in. Hypertension at early age. I never had any resistant hypertension, electrolyte abnormality, as Dr. Lazar mentioned. You have Kittelman-Liddle syndrome, have this hypokalemia, hypertension syndrome, sometimes not easily diagnosed clinically, but I actually have a few patients for that too. So this is the form that I give it to everybody. In the form, we just put like whether, it was ordered or not. So this is what I do for my office, right? Patient checks in the front office. And my medical assistant, she rooms the patient, and she gives the forms to the patient, and me or one of my doctor goes in there, we review the chart, and we talk to the patient as well. Sometimes if they have any questions, we just go and tell them why we think it's a genetic testing needed. And then MA at the end of the visit, MA, my medical assistant, collects the specimen. It's easy. We have I'm so glad Natera people gave me all the things, test kits in office. We just do the test over there. We put all the details and we FedEx it, and we're done with the visit. So it is possible within like 10 to 15 minutes of your visit. It's very easy. There is no obstacles in between. And then they go, and then when they come back, that's when we discuss about the results, which is going to take about 3 to 4 to 6 weeks, depending on that. So, and then it is also integrated to my EMR. So a lot of times, if the patient does not have appointment or they miss appointment, they can always look into the my chart, and they can look at the results, and they can always call me. I always tell them, do not get panicked, whether it's a positive or negative. Just talk to us before you get more problem or thinking about the genetic testing. So it's very easy in a regular office setup that you could do it. And this is when I did more, patients enroll, when I did that form and give it to everybody. We did more patients, of course. We did like 95 in 1 month and 73, we did a lot of tests. And then most of the, patient, actually 66% of the patient actually we did the Natera are met NCAF criteria. So some patients, they do want to just get it done, even they don't have any reason. They do, just for them, for them make to feel better that they don't have anything going on. So it's very easy to, get it done. Other one send that I want to put, like negative testing. Negative testing doesn't mean anything. It still means a lot, because sometimes a lot of genes that we don't have any thing right now, but they may get into later. So negative doesn't mean anything. It will give you at least the reasons that we're looking for is probably not there. There may be something else there. And negative also gives a assurance for the patient that they're not worried about the disease that what they're working or expecting for that. And also it helps the kidney donor. At least negative test will tell you that you're okay to go ahead and donate the kidney for your family. It needs to be done. So, and it it helps for the decision about if there's disease that we need to have any special clinical trial. So it's very easy to get it done. And this is other thing that I put in my slide before also. This is a template that I use in my office for every office patient. So the genetic testing is right there. So if I forget, it'll just give me another remembrance that, hey, I forgot the patient to have genetic testing done. So it's just easier. It gives me everything if I missed anything. It's just a thing that I practiced over time, so that I don't remember, I don't forget, and it's a very good patient practice. So I think, overall, I think genetic testing is a very routine tool that helps the patients to, diagnose and help for the family as well. Negative test still has a meaning of it. So, and I think there are a lot of genetic, counselors are helping if they have any questions. So it's very easy on a regular day-to-day office practice. So if you have any questions, please call me. And this is my email and phone number. Thank you so much for giving me the opportunity.
Thank you so much, Dr. Bhupali.
Thank you.
Why don't we, Lisa, let's see if we have questions from the audience.
Yes, couple of questions. First one, I believe, is from a patient. It says, I was lucky to get tested very early when Athena Diagnostics had developed early kidney testing in 2005, 2006 at Columbia University. I also met Tony and his growing team. Genetic testing seems obvious. Clinicians, please tell us why this doesn't happen. It seems so clear. What are the obstacles?
That's a really good question. Certainly I can tell you that during my training, I didn't learn just about anything about genetics. I was also an engineer in my first career, so my undergrad and then master's was really engineering, so it's not as if I had a lot of the biology, genetics undergrad. I hope the younger people than me are getting more general genetics, but somewhere around 2019 was where I felt, this is an area that I don't feel strong, and I started to see the articles in Clinical JASN, New England Journal, and recognizing this is suddenly a primetime issue in nephrology. So I still think that we have to educate ourselves. Luckily, companies like Natera are really out there trying to get people up to date and understanding the impact of doing genetic testing every day. And so I also think that we went from having almost no therapies and nothing new exciting to having like too many new things. So any given day I see someone and I'm still worried about hemoglobin A1C, blood pressure control, the LDL cholesterol, because, we think cardiovascular risk reduction first and foremost. But, I do a lot of intermittent fasting with people, and then I'm trying to enroll people in trials, and, how do you keep everything top of mind? So it's challenging out there, and we need to use our technology, our electronic health records, to try to identify people for whom automatically using like what Dr. Vipali just showed, using something like that electronically to trigger automatic genetic testing. Sadly, at times we have to leapfrog healthcare providers, sorry, to try to get people tested and provide the best care while we wait for them to kind of catch up.
Yeah. Next question will be for Maggie. In the past, cost and limited people doing testing. Is there a national list of genetic counselors with experience?
Yes, this is a great question. So there is so to get to the how to find a genetic counselor, if you go to the National Society of Genetic Counselors, and I can type that in the chat in a little bit, there's a Find a Counselor tool that can help you locate a genetic counselor in your area. To get at the specifics of, are there genetic counselors that are specific to renal genetics? It's becoming more common, but there aren't necessarily any. That's not to say that a genetic counselor, though, the tenets of genetic counseling remain the same. A lot of what we do is helping to coordinate the testing, explain inheritance, those sorts of things. So we don't necessarily have to be a complete expert in kidney genetics to understand and help guide a patient with a particular test result. I will mention that certain laboratories offer genetic counseling services. So, for example, Natera, which is my employer, offers pre and post test complementary genetic counseling services to patients. And then genetic testing, it's a myth that genetic testing is not affordable. The large majority of patients that have our Renasight testing pay $0 out of pocket. It's generally covered by insurance, covered by Medicare and so forth. So, and even those that do pay out of pocket, it's generally a very small amount. And we do have compassionate, and most laboratories offer this as well, compassionate pricing, so that if someone's able to provide some financial statement records and things, that testing may be offered on a sliding scale as low as $0. So that's something to be aware of as well.
Thank you very much.
Is that it, Lisa?
Yes, I think that's all we have time for.
Okay. Yes, I think that's probably good to move on to the next session. Thank you so much for a
Thank you.
very informative panel.
Thank you.
I'm going to type that Find a Counselor into the chat. I hope that will show for everybody. Okay.
And we will share that also when we do our written Q&As and share with the audience after the. All right. Our last session today features Peter Harris. He is a member of, our National Scientific Advisory Board and also at the Mayo Clinic. And he's going to share Mayo's streamlined process for diagnosing ADTKD and referring patients to the International Registry at Wake Forest University. Peter?
Hi. How are you?
I am great. Thank you for joining us today.
Just trying to share these things. Oh, let me put it onto the show a minute. Is that okay now?
Yes, we can see you and hear you fine. Thanks.
Okay. So I think I'm going to reiterate quite a lot of things you've been hearing about, the difficulty of diagnosing ADTKD without genetic testing. So the importance of genetic testing, importance of referring patients so that they are introduced into the registry. And at the end, I just do a little bit of comparison with autosomal dominant polycystic kidney disease. So right now I'm at a meeting of the PKD Foundation, a grants review session. So this is a kind of working at a parallel foundation right now. So I thought I'd start with just a couple of patients, just to reiterate the points I've been trying to make. This is one with kidney failure at 60 years of age. You can see here a strong family history suggesting dominant inheritance. You can see some complications coming with CKD. There's a few other things here that maybe you wouldn't expect in ADTKD, pancreatic cysts, a possibility of FSGS here, as well as tubular atrophy. And you can see the kidneys here, they have multiple cysts in, maybe more again than we'd expect normally in an ADTKD patient, but maybe this is because they're at end stage here. But you can see that the genetic testing identified a UMOD variant, and although this was initially called as a VUS by the testing company, we highlighted that this has been described before as a UMOD variant of significance, and so they upgraded to to a lightly pathogenic change, and the listing of variants at Wake Forest is very useful as a reference for this. Here's a second patient here. We can see, again, the declining kidney function at 35 years of age. You can see a few cysts within the kidney, of a large cyst here, for instance. The family history here is a little unclear, although there was some suggestion that the grandfather may have kidney disease. You can see that the kidneys are quite small, so not really large like we see in ADPKD. You can see the initial genetic testing using a genetic testing panel came back with a variant in a glomerular disease gene, but this is recessive and probably not important here. Likewise, a ciliopathy gene came up with a truncating change here. But again, this is a recessive disease, so not likely important, although some ciliopathy genes, such as IFT140, have been associated with a PKD-like phenotype. So maybe they could be associated with the cysts we see in this patient. But we didn't think that these fully satisfied or explain the disease that we're seeing in the patient here. So we sent to Wake Forest the Broad for MUC1 specific testing, and that came out as positive for the canonical C insertion that is seen with this disease. So if we think about how we deal with patients at Mayo in our virtual genetic nephrology clinic, so these might be patients that have a family history of CKD. So we typically refer them for a genetic testing here. This can be Natera testing with a gene panel, or variantics, which is again looking at kidney disease genes, but this time on a whole genome background. Both of these methods are good for identifying UMOD variants, but neither of them, as you've heard, are really good for finding MUC1 variants. So if they're negative from this testing, we refer for MUC1 testing to Wake Forest. Just a few things to think about here. Not all ADTKD patients are going to have a positive family history. Genetic results, you can come back as we saw in the second case there, can be quite confusing, suggesting other things that might be significant. And the presentation also can be rather confusing as well. As we've heard, it could be described as hypertensive related CKD or maybe diabetic CKD. And there may be other comorbidities, as we saw with that FSGS in one case. So I think genetic testing in a broad way for patients with a decline in kidney function and no clear example, and especially if there's a family history, is warranted. So what do we suggest when an ADPKD patient is diagnosed? So the local nephrologist is still going to care for that patient, and as we saw before, or heard mentioned, cascade testing can be very helpful in a dominant disease like this to try and identify other affected members. And as we heard also, this is the insurances will usually cover this type of testing, as it will the primary testing. Then we inform the patient and the family about Dr. Blyer and the Wake Forest team. We also inform them about the Rare Kidney Disease Foundation. We have some nice leaflets that they kindly sent to us to help with that. We've stressed the importance for clinical trials of the value of being added to the International ADTKD registry. And we can also supply, with the patient's permission, we can also give contact information to the Wake Forest group. So I think that the importance of genetic testing here, just want to emphasize ADTKD is obviously difficult to diagnose just on the clinical grounds. So genetic testing is definitely needed. In some cases, the specific MUC1 testing. The disease is fairly rare, although probably greatly underdiagnosed. So large populations that have been defined genetically are going to be important for doing clinical trials. We can, once we've identified one member, we can identify other family members, and this allows them to be known and so candidates for a future clinical trial. And likewise, the renal clinic that is seeing these patients may also be a site which will be able to host a clinical trial in the future. Finally, I just wanted to just do some sort of comparisons with ADPKD. So this is a much more common disease, around 1 in 1,000 individuals affected. In this case, the major genes were identified more than 30 years ago, although as you heard earlier, there are some minor genes still being identified. The first, and in fact, only approved treatment, tolvaptan, was approved around 8 years ago. So you can see that this was a very long period of time. I think the hope would be that it certainly would not be as long as this for ADTKD. And I think there's reasons for hope there in the development of the technology for ASO treatments, gene editing, also our better understanding of folding and trafficking drugs that have been pioneered for CF. Also, I think the pathogenesis of ADTKD is better understood than ADPKD. So there's now many therapies that are being tested either clinically or preclinically in ADPKD. Is probably 20 companies that are involved in these studies right now. I think one thing that's been important for designing clinical studies and clinical trials is to have large populations that have been collected and characterized. In this case, imaging is very important, looking at the size of the kidneys, the genetics, as we've discussed, and also longitudinal outcome data. And these have been key to designing clinical trials in ADTKD. And so I think this is another reason for referring patients to the International ADTKD registry so that we can get a better idea about the natural history of the disease, which is going to help the design of clinical trials. I think that's all I have to say, but thank you.
Thank you very much, Peter. And I'm mindful of the time here. And so can I request, Peter, that if we get questions from folks that we can send them to you and you can answer them and we'll post them after the summit.
Sounds great.
All right. Thanks so much.
Okay. Bye.
So you've heard a lot today about genetic testing, the importance of genetic testing for learning about this disease, for setting the stage for a clinical trial. We're going to hear more tomorrow about this International ADTKD registry. So please stay tuned for that and all the other presentations tomorrow. Before you log off Zoom, I would just request that there's a brief survey that will come up when we end the webinar. It'll take you 2 minutes to just fill out what you thought of this content, but also what information, what topics you'd like to see us cover next year, because we want this to be relevant and answer your questions and build a community and the understanding of this disease. So please take that brief survey. On behalf of everybody here at the Rare Kidney Disease Foundation, we'd like to thank today's panelists, all the physician researchers who continue to broaden and deepen our understanding of this disease, some of whom you've heard today, many more out there doing the good work. And we'd like to thank all the clinical nephrologists who are committed to improving patient care, which more and more involves genetic testing. All right, so I think we'll leave it there for today. Have a great rest of your day, and we'll see you back here tomorrow morning or afternoon for our friends in over the pond. All right, bye-bye, everyone.